Good morning, everybody, all my viewers. Today, I'm very grateful to present another question. I have two of them which I will present consecutively. I've already presented the data questions for the year 2001, 2002, and 2003. So today, I want to continue with the data questions, and I'm going to present the data question which occurred in KCSE Biology of 2004, which will be followed very soon by the data question of the year 2005. Remember, I present these data questions because they are like compulsory. In paper 1, they are compulsory. So you have to do the data question, then you select the other question, which is an essay. So, in this question, is a very interesting question, and I will open by saying the following. Have you ever wondered what happens inside a tiny cereal seed during germination? What if I told you that as it grows, some parts of the seed mysteriously lose weight while others gain? Let's explore this fascinating biological process and uncover the secrets behind the seed germination. By the end of the video, you will not only understand how a seed transforms into a growing plant, but you will also be able to explain the dynamics, changes in weight between the embryo and the endosperm during germination. This insight is key to mastering biology exams and understanding plant development on a deeper level. As a senior biology examiner, I've seen this topic appear in numerous biology exams. Mastering it could make all the difference in your understanding of plant growth. Whether you are studying for exams or just curious about plant biology, this video will equip you with the knowledge that goes beyond textbooks and helps you excel in your exams. So, welcome, guys, to Biology Paper 1, 2004 data question was question number 17, and it was giving you a score of 20 marks. Welcome, guys. The question was framed as follows. During germination and growth of a cereal, the dry weight of endosperm, the embryo, and the total dry weight were determined at 2-day intervals. The results are as shown in the table below. So those are the results. The results are there. Time after planting days, 2-day interval 0, 2, 4, 6, 8, 10. Dry weight of endosperm in milligrams 43, 40, 33, 20, 10, 6. Dry weight of embryo 2, 2, 7, 17, 25, 33. Total dry weight 45, 42, 40, 37, 35, and 39. So you are supposed to using all right, let me set this thing, guys. There's something which is not legible. I'm trying to set so that you can be able to read it properly. I don't know what is wrong with my setting here. There's a problem. Okay, using the same axis, draw graphs of dry weight of endosperm, embryo, and total weight against time. You get 7 marks. What was the total dry weight in day 5? Account for decrease in dry weight of endosperm from day 6 to day 10. Account for increase in dry weight of an embryo from 0 to day 10. Account for decrease in dry weight from day 0 to day 8. And you account increase in total dry weight after day 8. State factors within the seed and the 2 outside the seed that cause dormancy. Within the seed, outside the seed. Give the characteristics of meristematic cells. To plot the graphs of dry weight of the endosperm, embryo, and the total dry weight against time, follow these steps. Step 1, prepare the data. In this case, our data is already prepared. It's already in the table with the columns for time, dry weight of endosperm, and dry weight of embryo, and the total dry weight. We have already given it above there. Select graph axis. X-axis horizontal will represent the time in days. Y-axis vertical will represent the dry weight in grams or milligrams depending on the weight. Step 3, plot the data for each time interval, that is every 2 days. Plot the corresponding values of the dry weight of the endosperm, embryo, and the total dry weight on the y-axis. Use different markers or lines to represent each component, endosperm, embryo, and the total dry weight. Draw the graph. So there are 3 graphs. Endosperm graph, you plot the dry weight values of the endosperm against time. Embryo graph, you plot the dry weight values of embryo against the time. Total dry weight, you plot the total dry weight of values against time. Using different colors or line styles, solid dashed, to distinguish between the three data sets. Step 5, label the graphs or identify the graphs. Label the x-axis as time, days, the y-axis as dry weight, G or milligrams, and a key or rigid to distinguish between the endosperm, embryo, and the total dry weight of the calves. Interpret the graph. The dry weight of the endosperm typically decreases over time as it is consumed during germination. The dry weight of the embryo increases as the plant grows. The total dry weight might decrease initially due to utilization of stored food in the endosperm, but it should eventually stabilize or increase as photosynthesis begins. This is just our interpretation. We have not yet seen how our graph will look like, so continue, my dear friends. There is a one there we have used rigid. A legend in graphs is a key that helps to identify and explain the various symbols, colors, patterns, or lines used in the graph. It provides information on what each part of the graph represents, making it easier to understand the data being presented. For example, in a bar chart, the legend might explain which colors correspond to different categories, or in a line graph, it might indicate which lines represent different data sets. In short, the word legend helps to decode the visual elements of the graph for clear interpretation. So, graphs of dry weight of endosperm, embryo, and total dry weights are there. Okay, that's the graph. You can see x-axis the time days, y-axis dry weight milligrams. The graphs are 3: identity, total dry weight, embryo, and endosperm. So this is one way of identifying the curves. So how do you want the mark? Axis gets 1 mark, scales 1 mark, plotting 3 marks because there are 3 graphs, identification 1.5 marks, labels on the line graph, curves or smooth curves 1.5 marks. The total marks is 8 marks, maximum 7. So you can see the marks have been put, like you can see on your y-axis, dry weight have a mark, time days have a mark, scale, x-axis have a mark, y-axis have a mark, plotting, you can see the 3 P's there, P, 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 a tick, curves, you can see the half, 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 and then you can see the identification, a half, half, half. All right, so that now the total mark is 8, but you give maximum 7. Now, this is another way you could have drawn the graphs, guys. The graphs could also have been drawn that way. All right. Okay. Dry weight of endosperm embryo and the total weight over time. So in this one, you can see we have 3 colors representing the graph. We have red for total dry weight, blue for endosperm, and green for embryo. All right. So you can see time after planting, days, dry weight, milligrams. Okay. So these are 2 contrasting ways of how you can draw your graph. Okay. And how you can identify. So for this one with the colors, you just give the legend, which is there at the top right-hand corner of this graph. And you have a smart graph. Look at the plotting in this one. Here we have used the dots. It is around in biology, although mathematicians will not agree with this, but in biology we can put the dots. All right. In the previous one, we used X or cross to mark a point. Okay. All right. Key observation: endosperm weight decreases as time progresses, indicating it is being consumed during the growth process. Embryo weight increases steadily as the plant develops. Total dry weight decreases initially, but increases later as the embryo grows and photosynthesis likely begins. So let's go to the questions which were asked in the examination. What was the total dry weight in day 5? You are given 1 mark, and you read from the graph, it is that 8.5 milligrams plus or minus 0.5. So it can be 39 or 37. Account for decrease in dry weight of endosperm from day 6 to day 10. Decrease in dry weight of endosperm from day 6 to day 10. Hydrolysis of starch into simple sugar, which are translocated to the embryo. Respiration of simple sugar to release energy or to release gaseous exchange material or to release heat. Increase in dry weight of an embryo from day 0 to 10. Here, the reason is new materials are being synthesized from protein, bringing about growth of the embryo. Decrease in total dry weight from day 0 to day 8. The reason is the rate of respiration is faster than that of synthesis of materials for growth. Increase in total dry weight after day 8. Reason, the first forage leaves carry photosynthesis leading to growth. State 2 factors within the seed and 2 outside the seed that cause dormancy. So within the seed, we have presence of abscisic acid, or you can call it germination inhibitor. Again, embryo not fully developed, and also absence of hormones, all enzymes that stimulate germination. They are known as gibberellins, cytokinins, and finally, the cause of dormancy is impermeable syndicate. So how many causes can you see there? But you are choosing only two. We have presence of abscisic acid, embryo not fully developed, absence of hormones that stimulate germination, impermeable syndicate. Outside the seed, low temperatures, absence of light, and lack of oxygen, lack of water. So from any two of those, you get your 2 marks. Give the characteristics of meristematic cells. 1, they have a dense cytoplasm. 2, they have thin cell walls. And 3, they don't have vacuous. So there is absence of vacuous. So we come to the end of that video by acknowledging the team which inspired me to come up with this video. I also thank my family members for being patient with me, because there are times I'm typing and concentrating, okay? Sometimes I have to not take a meal because maybe I was in the middle of typing. They are patient. I won't forget to thank my teammates in the neck marking troop who participated in coming up with this final draft when the senior examiners met for modulation of the marking scheme. Finally, I won't forget the ChatGPT, which is an AI language model developed by OpenAI, because it is providing assistance by creating and polishing some of the content for this video. So my dear viewers, that brings us to the end of the way the data question of 2004 was supposed to be tackled. I hope you watch the video many times to internalize these important biological processes. So goodbye and see you in my next video.